intimidating at the same time, because I really honestly remember the first seminar that I ever went to was in this room, and it was Colin Trevathan who was giving it, and it was about babies and development and language, and I was blown away. Like, that was such a turning point for me in my, like, oh, this is amazing. I could do this sort of research, and it just feels a very pivotal moment for me. So I can't say too much more about my years as an undergraduate here, because they were not successful, and I was a pretty awful student, but it was, it can work out. You can see the light, but it took me a while. So I was a student at Christ College here in Cambridge, and I did natural sciences, and I originally wanted to do geophysics, so I was doing the physics stream, and then I did very badly in that. And my director of studies suggested maybe biology was really where I kind of lay, and I thought psychology looked good, and then I did zoology, and I did history and philosophy of science because there were no practical classes. That was my, having spent Saturdays in the Lensfield Road, it's always called the chemistry labs, doing, Saturday labs. I didn't want to do any more practicals. So anyway, psychology was, like, for me, really exciting, and I felt really lucky that I was doing a degree that allowed me to change and make this choice, and it's been really, a successful one for me, I can say. So after I graduated here, I went to London, and I was working as a research assistant at Great Ormond Street Hospital and the Institute of Child Health, which is part of UCL, and doing neuropsychological assessments with children who had brain damage. And brain imaging was in its infancy. It was literally just starting then. So I was quite lucky to be around at that time. And what was really interesting to me was that I was seeing these children who were having quite large removals of brain tissue because of epilepsy that was intractable to drugs. So this was the only recourse they had for treatment, and it was a successful treatment in treating their epilepsy. But these kids could have whole hemisphere removals of even the left hemisphere and yet still develop normal language. They required a certain set of sort of circumstances for it to be normal, but it wasn't stuttering, it wasn't developmental language disorder. The kinds of difficulties they were having were very different, really minor, I think you would say. Quite honestly, these kids were doing really well. And then in contrast, I was, for my PhD, which I was doing part-time, working with this family known as the K family, who had a really severe language impairment. There would be no question if they were here now that you would be able to say that person has a language impairment and that person doesn't. You don't need to do assessments to work that out. And yet their brain images were normal. So there was no obvious lesion, there was nothing on the MRI scans that you could say would explain why these individuals had a language impairment, and those that have a really large lesion, like a whole hemisphere removed, have no language impairment. So this was my kind of puzzle. Not long after we'd worked with this family, the gene mutation that explains the deficit that the members of this family, the K family, have. Can you see my? Yeah, okay. I'll put this on. Was discovered as a gene called FOXP2, and the discovery of that gene's kind of led to a proliferation of research in genetic and animal models looking at how FOXP2 affects speech traits in other species. Obviously they don't speak, but looking at bird song development, also communication in bats, for example, and the effects of FOXP2 abnormal variants on that kind of auditory, motor, vocal learning, if you like. So the differences that I then found using some quantitative measures of MRI scans that look normal were very subtle differences in the amount of tissue that was in the caudate nucleus. And the caudate nucleus is part of the basal ganglia or striatum. It's a subcortical nucleus that you may know is affected in patients who have Parkinson's disease. So largely we attribute kind of motor functions and motor learning to these subcortical areas, not typically areas you would associate with speech and language. But if you think about it from a sort of domain general point of view, that being able to learn to associate auditory and motor information might rely on these more general kind of learning circuits. So that was kind of, I've explained it in that level of detail because it kind of sets the scene for the research that I'm going to talk to you about some 35 years later. It's taken me that long. But after I did my PhD at UCL, I then took, I went to Canada, to Montreal, to the Montreal Neurological Institute to do more brain imaging work, and there I learned to do brain stimulation as well. So I was there as a postdoc for 5 years, and then I came back to Oxford working in the Brain Imaging Centre in Oxford in 2003, and then I got this job in the psychology department where my project supervisor from here, Oliver Braddock, was the head of department. I'm sure that's not why I got the job, but it was a nice kind of circularity to my academic journey, and I am now been there a long time, and I also work at St Anne's College in Oxford. Okay, so just to revisit the kind of theoretical framework for this talk, to remind you, so children who have focal lesions acquired early in life, that's necessary, right? It has to be early, probably before any cortex has become dedicated to language processing. They can develop good language even if it's involving the whole of the right hemisphere. And children who have developmental disorders of speech and language don't have frank neurological lesions. If they do, we rule them out we understand the cause of their difficulties, so we rule them out of these classic definitions of disorders. We think there's probably a genetic cause of these disorders, and we know that's true in some cases. And that genetic etiology is going to not just affect one hemisphere at a time or one circuit, but it's going to have a more diffuse effect probably on neuronal development and affect perhaps networks that exist in both hemispheres, not just tied to language circuitry in the left, say. And it's possible using now even more sophisticated brain imaging methods for acquisition of data and analysis of data to detect these quite subtle kind of differences in the brain structure and function. And I'm going to give you some examples of that. And I want you to bear in mind when I give you these examples that not all of them these differences that we can detect might cause the language impairment or the speech impairment, but actually some of them might be consequential to it. So the experience of having a language difficulty is going to have impact on the brain as well. So my framework goes back to that finding in the K family that the caudate nucleus was involved in this kind of speech motor disorder that they had. So we think it's likely that the genetics, affect FOXP2 in that case, have affected the development of these circuits that come from the cortex into the striatum, here shown in red and green. And these are sort of parallel circuits that then output back up to cortex via the thalamus. So I might use these words interchangeably. Hopefully I'll be more specific, but I'll use probably basal ganglia and striatum interchangeably. But by striatum, I mean the caudate nucleus and the putamen, which are the input nuclei to the basal ganglia. So cortex projects to those nuclei in a topographic way. So the motor cortex, which is in the middle of the hemisphere here, projects to the putamen, which is sort of in the middle of the subcortical area. The frontal cortex, association cortex, projects to the caudate nucleus, over here, putamen over here. Caudate over here is a bit more dorsal and a bit more further forward in the brain, so it gets its input from all of the cortex, but primarily we're talking about prefrontal cortex. So just in a very kind of simple dichotomy, you might expect a disorder that's more motoric in nature, like a speech disorder like stuttering, to have effects that we can see in the circuits through the putamen, whereas a disorder that's more language-based, has a bit more of a cognitive component to it, would affect these circuits that come via the caudate nucleus instead. And there's this other circuit often referred to simply as this limbic circuit, which I don't particularly like that term, but involves the ventral striatum, which is responsive to sort of rewards and emotional processing in the brain. And many of the kind of factors that we measure in children or adults with developmental disorders of speech and language are impacted by other kind of factors that I think probably reflect the inputs to these ventral striatal circuits that sort of modulate things like emotions, perceived threat of some sort of conversation, or the reward that you might get typically from a learning situation, from reading, enjoyment, that sort of thing. So there's a lot of modulation of these very simple circuits by other circuits that I'm not going to talk about, but just want you to keep in mind. Okay, so I'm going to talk about speech, and that'll be the work on developmental stuttering that I've done. I'm going to talk about brain stimulation study, some functional imaging, and some recent imaging looking at iron in the brains of people who stutter. And then I'll talk about language. This is a study with children who have developmental language disorder, which I'll explain later, looking at the macrostructure of the brain, some of the more quantitative elements that we've got, looking at how much myelin there is that affects the conduction of information in the brain, and then some learning circuits. Okay, sorry, that's a long introduction. So this is a set of pictures that I like to show, especially when I'm talking to children about famous people who stutter or stammer. Again, I'll use those interchangeably. So stuttering affects between 5 and 8% of children. And at that point, there are typically 2 males for every female child who stutters. And most of those children, 80% of them, will recover from stuttering or stop stuttering during childhood, leaving us with an adult rate of 1%. And at that point, we have 4 or 5 males to every female who stutters. So it looks like this spontaneous recovery or stopping of stuttering is more available to female children than male children, and we don't understand that. There are genes that have been identified that are causative of stuttering, and we know from brain imaging studies, including my own work, that there are neural differences in the brains of stuttering. So that's a big piece of work. You might think sometimes your own work isn't that relevant, but just telling people who stutter that it's not their fault, it's not because they're nervous or they have this disposition, that actually there is a neural difference, a sort of cause. For some people, that's information they're very happy to receive and to be able to share. So, yeah. So lots of these people on this screen I've picked because they have lovely voices, like James Earl Jones, who was the voice of Mufasa and Darth Vader. Then we have Mr. Bean, or what's it called?
Rowan Atkins.
Rowan Atkins. Thank you. Ed Balls. We've got singers, Stormzy, or rappers. Ed Sheeran, Emily Blunt, Joe Biden. Lots of sports stars. So Shaquille O'Neal, James, who plays football for Colombia, I think. Another footballer there. So, you know, this is really just to emphasize that it does seem to be perhaps a motor system difference that is limited to the speech motor system, not necessarily affecting other motor abilities that would lead you to be a better sports person or a musician, for example. Theoretically, there is a lot of research that has described why the basal ganglia or dopamine or both are affected in stuttering and give rise to stuttering behavior or the kind of motor symptoms that are characteristic of stuttering. But actually, the imaging evidence to support that theory is a bit limited. So there are a few studies that have found differences in brain activity during speech in people who stutter, which is what PWS means on these slides. But actually not much. And I actually think there might be a really boring reason for that, which is that a lot of the brain imaging studies that are done these days are done at 3 Tesla, which is a much higher field than we used to use in the old days, at 1.5. And basal ganglia signals are not well detected when you're imaging oxygen levels, which you need to do for fMRI in that region. So the data from, and then some of the data I'll talk about later might also explain why I think this might be specific to stuttering as a problem. But needless to say, I've looked very hard to find differences in activity in the basal ganglia in people who stutter, and I've not found it. There is also a lot of theory based on Parkinson's disease and the similarities between stuttering and Parkinson's disease. Also, patients who have Parkinson's disease can develop stuttering symptoms, or these may have re-emerged from a developmental disorder that went away. But then you have, so dopamine is typically depleted in Parkinson's disease, but we know that if you then block dopamine in people who stutter, you can improve their fluency. And then we know if you give them L-Dopa, for example, more dopamine, then their stuttering gets worse. So there are really mixed effects in both directions, too much, too little dopamine. And I mean, that's quite typical of dopamine. There seems to be like a sweet spot that you need, but it's not clear what role dopamine might play in stuttering. But one of the things that's relevant and interesting is that Parkinson's patients can benefit from cueing, from external cueing, either using music like a beat, or stripes like on a zebra crossing or on a stairs as visual cues to initiate movement. So you can enhance gait fluency in Parkinson's disease by external cueing. And you can do the same in stuttering. So you can make someone who is typically disfluent completely and immediately fluent by giving them an external cue to initiate speech. So you can use a metronome, for example. There are lots of ways of temporarily, so you take the cue away and stuttering returns, right? Temporarily enhancing fluency. So you probably have seen on these shows that there is often someone who stutters, but when they sing, they're completely fluent during singing, and then they stop singing and they still have problems with speech production. If you saw the King's Speech film, they used all of these techniques here. So you can mask speech with noise. You can't hear yourself. That induces fluency. Just altering feedback with a delay or a frequency shift or other kinds of sensory feedback all induce fluency. Or you can ask people to change the typical manner in which they produce speech. So move from some habitual way of speaking to more effortful, different way of speaking. So adopt a different accent, or speak with as if you're on helium, or like Mickey Mouse, or just whispering. All of these completely and utterly get rid of the disfluency, and they act immediately. You don't need to learn them. But people don't like to use them because they're quite effortful, and they'll say, oh, I sound funny if I speak like Mickey Mouse. I don't want to sound, you know. Another method that we've used, and I'll give you some examples in the talk, are asking people to speak in time with an external cue. So we use a metronome, or read or recite something in unison with someone else. So that's choral reading. And again, both of those are fluency enhancing. So with this in mind, we decided to use these fluency enhancers in a randomized controlled trial to treat people who stutter using brain stimulation to enhance this fluent state. So we put people into these temporary fluent states by using these fluency enhancers, and then we apply a very weak electrical current across the brain to basically promote that kind of speaking and help the brain to kind of learn this more fluent way of speaking. So we chose this method based on what we know from other studies where they've used these to look at motor learning. So this is a very difficult task. We have to squeeze this manipulandum and move it between these different bars. And people were given this task to do, and those that had the brain stimulation, so the anodal group here, showed much more rapid learning than people who didn't have stimulation. But importantly, after they'd done this week-long training, 90 days later, they still showed the benefit of this learning with stimulation. So lots of studies, I've picked a very good one here, but there are lots of studies showing how this very sort of cheap and fairly safe, or it is safe, I think it's very safe, form of brain stimulation can promote learning. I don't think there's anything sort of magic about TDCS or other forms of brain stimulation. I think they just give you rapid games that really help you to keep going with the kind of practice that you're getting on the motor learning task. And the motor learning task in our case was speaking fluently. If we could prove this worked, this would mean that we could do this quite safely. People can have these kits at home, but speech and language therapists could have them in the clinic. They're quite easy to use. They're quite easy to train people to use them. But they're only really going to work if you've got a good therapy or a good treatment that it can promote for you. So we did this randomized controlled trial where we allocated 30 adult men who had moderate to severe stuttering into 2 groups. They were blind and the researchers were blind to the allocation. You can do that with the TDCS box. You can set it up to do a stimulation that's real or sham without you knowing. And they had 5 days of 1 milliamp, that's very weak current, applied for 20 minutes while they were put into this temporary fluent state using one of these techniques. And then we saw them again 1 week and 6 weeks later. So we pre-registered this with the clinicaltrials.gov website. So the induction involved some speech samples every day. So the intervention involved speech samples every day. They then had 4 tasks that we thought were a bit interesting to do, more than just doing the same thing for 20 minutes. So 4 5-minute tasks, reading live in chorus with the researcher, reading a bit harder in chorus with an audiobook, narrating a video with a metronome to keep time, and then having a conversation with the researcher using the metronome to induce fluency. And I'm just going to show a video of one of the participants doing this. So you'll see, first of all, his speech without any fluency induction, and then you'll see him speaking with the fluency induction. He's wearing the electrodes, but he's not actually being stimulated. So, we saw them at 1 week and then 6 weeks after the end of this week-long intervention. And what we found was that there was a significant reduction in the so this is change in percentage disfluency. So the number of disfluent syllables that they produced, the baseline averages were 12%. After these 5 days of 20 minutes a day training, we saw a reduction of between 3 and 4% in that. So it's I mean, that sounds small, but it's only 5 days and it's a quarter symptom reduction. The sham group, who had the training but no brain stimulation, showed no change from their baseline levels. We didn't expect to be sensitive enough using this clinical measure, which is called the stuttering severity instrument, to detect a change, but we did. So you see this reduction after 1 week of about 7 points on the SSI, and 6 weeks later it's gone back closer to baseline, so they're not maintaining this necessarily, but it's still different from sham. And you've probably noticed that at 6 weeks after the intervention, we're still seeing an effect, but it's limited to the reading samples and not to the conversation samples. So the conversation disfluency has gone back up to 0. On the whole, it's not true of everybody. People who stutter tend to show more stuttering on reading samples than on speaking samples, mainly because I think if you're very practiced, you can choose to avoid words that you know are going to make you stumble, whereas if you're reading, you don't have that choice. You have to read what's before you. Okay, so we also scanned these people using MRI while they were reading sentences out loud in the scanner. So nobody could read, I think this is Farsi. So that was our baseline kind of visual control. We got rid of any stuttered samples in this analysis. So all the analyses are of fluent sentences. And we are comparing the brain activity evoked by reading a sentence out loud in the scanner with before the fluency induction with after. But again, for fluent sentences in both times. And what we see when we do, there is that there is an increase in the bold activity, fMRI activity, in the putamen and in speech motor cortex. So this is the kind of classic Broca's area, inferior frontal gyrus and anterior insular, a little bit of temporal cortex here. You can see the insular again here, and then this is the putamen. So this interaction that we're seeing here means that there was no change in the bold signal in the sham group in this area, but that the group getting anodal stimulation, real stimulation, increased activity after the successful intervention compared to before. Does that make sense? So for the first time, we're finding differences in the putamen, a lower threshold on the left as well. This is in the right, in this group. And so that also fit with some other work that had been done in Japan, where they used metronome timed speech alone for a very intensive training period of about 8 weeks in people who stutter, and they also showed an increase in activity in the putamen in that group. So as I say, the brain stimulation isn't necessarily essential for this, but it kind of brings about effects more easily and more quickly, and that in itself kind of maintains people's engagement with perhaps the training. Okay, recently in a completely separate study of people, adult men and women who stutter, we used this sequence which is called sometimes histological MRI, but it gives you quantitative data from a reasonably short sequence of 20 minutes that is sensitive to the amount of myelin in the brain. So that's the white matter, fatty substance that coats axons and improves the conduction of neuronal signals between areas, and also iron. So you have more iron in your globus pallidus in your brain than in your liver per tissue volume. So there's a lot of iron in the brain. We were interested in this sequence because, as you can see, you get good sensitivity of measurements in the basal ganglia here, even if you're looking at myelin in grey matter. But we didn't have necessarily a directional hypothesis as to whether people who stutter would show more or less iron. We were just looking for a difference in the signal in these structures. So I feel like this result was a little bit of kind of luck in some ways. But what we found was that people who stutter had significantly more iron concentration in the putamen, again shown here. And again, this left lateralized set of areas in speech motor cortex, the insular, inferior frontal gyrus, etc. And when we look at, again, this pattern is bilateral if you lower the statistical threshold, but it's higher on the left. If you look at the data, if you extract all of the values for each individual in the study, from the fluent group in black and the stuttering group in red, and plot that with age in our data, then you can see that both groups, as expected, increase the amount of iron in this structure and in the cortex. That's typical, but that the people who stutter had more iron to begin with, even at the lower age, at the youngest ages in this study. So we're seeing more iron here. So this is a reasonably big effect size. It's interesting for other reasons, because I told you there were some genes that had been identified in stuttering, and we can now link those genes to this story about iron. So before it was a bit kind of like, what do these genes do and why are they implicated in stuttering? This provides some kind of link. I think it would be really important, we haven't done this yet, to see at what age in development does this iron difference occur, and is some sort of iron homeostasis the cause of stuttering persistence in development? Is that why some people persist and some people don't? Is that why there's a sex difference? Do women have less iron? They certainly do at during puberty, for example, but recovery is typically earlier than that. Anyway, that's the end of this stuttering kind of set of studies that I wanted to talk to you about. Just to remind you, I've shown you that 5 consecutive days of TDCS with a temporary fluency induction produces longer lasting improvements to fluency, and other people have replicated these findings since we published that study. And we found with MRI that this change brings about a sort of normalization in the activity in the basal ganglia and in speech motor cortex. And we're finding in similar regions that where we see these changes in activity, that there are differences in the amount of iron in the brain. And I said I would explain why I think that the sensitivity at 3T is related to stuttering in particular. So iron will distort the T2 star signal that we measure when we're measuring bold signals. So if you have too much iron, you're going to have more disruption of the signal that we're measuring with bold, and that might be why we've failed to be sensitive to differences in signal in that region if the iron story holds here. Children, even if they have too much iron relative to control groups, still don't have that much iron, right? It's something that accumulates. You don't have any at birth, so you accumulate through life. So the differences that have been found in the basal ganglia have actually been mainly limited to studies of children who stutter. So there's a lot to kind of pursue in that area next, I think. Okay, shall I continue? So I was going to move on to developmental language disorder. So has anyone heard of this? I know somebody has. But pretty much it's the Cinderella of neurodevelopmental disorders, right? There's very little funding into it, very little public awareness of this, and yet it affects probably up to 2 children in every classroom of 30. So 7.5% of children have developmental language disorder. And what is it? Well, it's a relatively hidden disability that affects all aspects of language. So listening, speaking, reading, and writing. I think in our experience, it's often misdiagnosed as dyslexia. So children will be getting support in schools for dyslexia, but maybe they've got a language difficulty as well as one that's affecting their literacy. It has a lifelong impact, and it affects, academic achievement, employment opportunities. So it has big impact on the individual, on their families, and on society. And we know that there are genetic causes in probably up to maybe 70% of individuals with DLD. So I'm going to call it DLD, developmental language disorder, for the rest of the talk. In 2016, we wrote this paper which summarized the kinds of language learning difficulties that children with developmental language disorder have, and made the claim that actually most of the tasks on which they were impaired were tasks that fall under the kind of procedural umbrella of non-declarative learning, if you like, or implicit learning, and they were typically sequential tasks. So ones that were non-sequential, like probabilistic learning, there was one paper that showed impairment there, but others that showed that children with developmental language disorder, it used to be called SLI, specific language impairment, were typically intact on those types of tasks. So this was not something, this kind of classification or pointing to the deficits that explain developmental language disorder as being kind of skill-based, implicit or procedural in nature was not a sort of novel idea. This is something that Michael Ullman had written about extensively and has a theory or hypothesis called the procedural circuit deficits hypothesis. That was quite hard to say, tongue twister, that makes that claim that it's these, again, this sort of domain general language learning circuits that go via the striatum and are important for perhaps more implicit learnings of statistical sequences, of skills that are the core of the language learning deficit in a disorder like DLD. I think you probably don't need me to remind you about HM and this classification. So yeah, so this is Michael Ullman's hypothesis that he first described more than 20 years ago, but recently in Nature Human Behavior, they had a sort of meta-analysis of studies that had been done in DLD, and they found that 100% of the anomalies described in brain structure were described in the striatum. They call it the anterior neostriatum, which is this red area here, caudate and putamen, in developmental language disorder. And then something like 82 or 83% found differences in the frontal cortex in populations of children with DLD. So before this paper was published in 2024. In 2018, we started a study called the BOLD study, the Brain Organization in Language Development study. Turns out there are lots of BOLD studies in the world, so we had to be more specific and say, my favorite was something like bovine, something to do with bovine lung development. And, so we called it Oxford BOLD study, and we recruited 175 children from April 2018, and we stopped recruiting in January 2020. Lucky were we. So our inclusion criteria were that children were aged between 10 and 15 years, and they had normal hearing and had grown up in the UK speaking English from before the age of 5. We excluded anyone with a non-verbal IQ below 70, or if they had ADHD or autism spectrum disorder. So they had to have a frank diagnosis of that, not just score high on a scale. And mainly the reason for excluding these, because these are common co-occurring deficits, right, was that we didn't think they could lie still in the scanner for an hour, because that's how long we scanned them for. And then, the ASD, in our previous experience, which had some issues about feeling comfortable about consenting, some of the children, whether they really understood what they were consenting to. We gave them a bunch of language tests looking at expressive and receptive language for vocabulary, sentences and narrative skills, reading, verbal memory, motor skills, working memory, non-word repetition, which is a test that's pretty sensitive in DLD. And from that, because there were so many tests, we had a pre-registered factor analysis that allowed us to extract 2 factors that explained the variance in the data. One was a language factor and one was a memory factor. So to summarize all of these variables, we had these 2 factors. And that allowed us to do 2 kinds of analysis. We could either classify the children into categories based on research criteria for developmental language disorder. That would mean they had to have a history of speech and language problems, and they had to have scored below 1 standard deviation from the normative mean on 2 or more tests. That's a pretty standard criteria for this age range. Or we could use the language factors to look continuously, the language and the memory factors. So here you can see that actually the groups, so we have 2 groups typically developing in blue, DLD in yellow, that could be more or less separated on 1 factor, which is the language factor. And the memory factor helps a little bit more to break these 2 groups apart. In green, we have this 3rd group, which we call HSL, standing for History of Speech and Language. So these were children who either a parent or a teacher or a clinician had been concerned about them. They may even have had a previous diagnosis of DLD, been having support at school, but on the day of testing, they didn't meet our criteria. So for most of the analyses I'll explain today, I'm treating the groups as 2 groups of DLD and typically developing and ignoring the HSL, but in all of the work we've published, we've used the whole group as well to kind of confirm that if we just treat the data continuously, we see the same pattern of results. So of the 175, we had to get rid of data from 19 for various reasons, leaving us with 74 typically developing children, 54 DLD, and 28 with this HSL diagnosis or grouping. What do they look like? So remember, to get a DLD kind of classification in this study, they had to be impaired on at least 2 tests, lower than 1 standard deviation from the mean. And if we look at all of the data from all 50, so each of these little cells in this in these blocks shows you what their score was roughly in terms of z-scores or standard deviations from the mean. And you can see that the most sensitivity comes from this expressive test of sentence production. So they're asked to recall sentences, and only the receptive test we used here is the TROG, test of reception of grammar, and only 3 children out of the 56 were not impaired on one or both of these tests. You can see that the vocabulary, especially receptive vocabulary, is actually almost an area of strength in this group. And the narrative test is somewhere in between. And as I think is typical, it's more common to see expressive language problems rather than receptive language problems in this group. Okay, so we've published quite a lot of data on this study. We used that quantitative MRI sequence. I'm just going to give you some snapshots, and looked at myelin in this group and found that there was significantly reduced myelin in the grey matter in the caudate nucleus in the group of children with DLD. And we see that bilaterally here in the caudate. In the cortex, again, we see this sort of left lateralized pattern of results. And one idea here, thinking that the cause ultimately of these difficulties might be genetic, would be that the genetic effect would be pretty general, affecting maybe circuits of both hemispheres through the basal ganglia. And the reason we see this lateralized pattern of difference might reflect some sort of experience-dependent change, that they're not using language as much, they're not exposed to as much print, they're not reading as much as typically developing children. We wanted to test the hypothesis from the Ullman paper about the procedural circuits deficit hypothesis. We've shown difference in myelin in the caudate. We measured the volumes of the caudate nucleus of putamen and the hippocampus and the amygdala, as well as other subcortical nuclei, and we find that nearly all of them are smaller in children with DLD than in controls. Doesn't matter whether you correct for intracranial volume, which isn't different between the two groups, but you still see this deficit of a reduced volume of the striatum that fits with the prediction from the Ullman theory, but he would say that the hippocampus is unaffected, and these declarative learning systems for declarative memory can even compensate for the non-declarative impairment. And our data would suggest that that's not something that's going to be available to children with DLD. We also found differences in the cerebellum, so neither of the strain in different language circuit altogether. So specifically on the white matter tracts that bring signals from the brainstem, particularly like from the inferior olivary complex, which processes auditory information into the cerebellum. That was reduced in what's one of our measures from diffusion imaging, FA, in DLD. So this means that the connectivity isn't as good in DLD as it is in the typically developing group. We also found that there was an interaction between the age of the children and this measure of white matter integrity in the cerebellum, in that the older the children were, the lower their FA measure was here if they had DLD, but it went up with age in the TD group. So again, this looks like maybe some experience-dependent changes that's getting worse in these teenage years. Last slide on data, I think. This is just looking at cortical thickness and cortical surface area in the same children. And we found maybe what you would expect in the surface area in Broca's area and its homologue in the right hemisphere were reduced in children with DLD compared to typically developing children, and also this area in the ventral occipitotemporal cortex or fusiform gyrus, where is the sort of location of what we think is a reading specific area. The visual word form area would be located here in the left hemisphere. So again, for me, I haven't tested this hypothesis and don't have data that would allow me to do that yet, but for me, this could be something that has failed to mature because of the difference of experience in children with DLD, because they're not reading as much and they're not exposed to as much print as typically developing children. Okay, it was quite a whirlwind, but I have 2 more slides. So just to summarize the DLD half, I've shown you that the sentence processing tasks seem to be most sensitive to behavioral differences in DLD. That we see less myelin in the brains of children with DLD, particularly in the caudate nucleus bilaterally, that fits with the idea of this more cognitive circuit, if you like, being affected in this language disorder. The volumes of the subcortical nuclei are definitely smaller in DLD. That fits with the prediction from this procedural learning deficit hypothesis, but we find that the deficit is not limited to the striatum, and we even see things in cortical areas, but in the hippocampus and the cerebellum. So you could take a very pessimistic view of this and say, oh, everything is affected in DLD and it's all different. There's nothing specific about it. And I wouldn't have a very good argument against that, to be honest. Okay, so just to come back to my initial framework, I showed you this figure at the beginning. And again, I think that I've just used this framework to show you support for this idea that these circuits might be distinct and might be selectively affected in these different developmental disorders. And as I said, it's likely that you would have input from emotion processing and other cognitive effects that would further impact these circuits. Okay, so just to thank all of the lovely people who did all of the work that I've talked about today and our funders, and thank you for listening.